conference anyway. So the session 3 is all about new tools and innovation. So we'll be looking at how we bridge the gap between our current ancient trees and the ancients of the future. And first off, we've got Sonia Reveley here, who's the Woodland Officer at the Bat Conservation Trust. And she's going to be talking about the AudioMoth, which is an affordable survey tool and its future in bat monitoring. So Sonia, over to you.
Trust. I work with 4 of the Back from the Brink integrated projects: Roots of Rockingham, Dorset, Heath and Hart, Limestone Living Legacies, and of course the Ancients of the Future project. So for this talk, I'll introduce the AudioMoth, a sensor the size of a credit card that we've been using to survey some of our integrated projects' primary and secondary sites, some of which have never been surveyed before. And I'll summarize the approaches that have been taken by BCT in developing our PAM surveys for the National Bat Monitoring Programme, and we'll share the potential use of the data collected and where we're going with our passive acoustic monitoring at BCT. So monitoring bats in the woodland habitat has always been an ongoing challenge. There are currently 3 monitoring approaches that we use when surveying woodland habitats for bats. They are trapping, active acoustic surveys, and passive acoustic surveys. But they all have their own set of challenges. For active acoustic surveys, we need surveys that have a moderate experience of bat monitoring, and survey effort would generally need to be quite high in order to capture some of our rarest bat species, like our woodland specialists, are either typically rare or uncommon, or can echolocate quietly in order to sort of provide sufficient encounters to collect information to calculate things like population trends. With regards to trapping, this requires obviously a license plus experience, specialist equipment, and survey time. And for passive acoustic monitoring, bat species like Myotis bats are acoustically cryptic and cannot be reliably identified to species using their echolocation calls alone. Meanwhile, passive acoustic surveys do offer some advantages over active surveys and trapping. The sensor can be deployed in the field. They can be left unattended to record bat activity for long periods of time. And since really the AudioMoth was released, there's been upgrades to the firmware, which means that we can use bigger SD cards now to store more nights of data, and we can also leave the AudioMoth record for long periods of time. And this greatly increases the survey effort, while at the same time minimizing survey effort. As sensors are deployed during daylight, it's also easier to locate those sort of difficult to survey habitats, for example, woodland interior. And of course, it's safer for the survey because they can see where they're going. And it's also a technique that can be used with little or no previous experience of bat monitoring. So from my experiences with volunteers, we provided them instructions, and they've been happy to follow the instructions. They've been able to configure the AudioMoths, and they've been able to go out and deploy these sensors. Now, in the past, the use of passive acoustic monitoring for large-scale projects have been very costly, normally requiring detecting equipment that can retail at around £1,000 per unit. However, nowadays, with advances in technology and open-source design, we have affordable full-spectrum passive acoustic sensors like the AudioMoth here that you can see on this slide. And AudioMoths cost around sort of £60 per unit. And this means that we can deploy a number of AudioMoths, so an array of AudioMoths on one site to monitor for bat activity. And this has allowed BCT to think about biodiversity monitoring, obviously on a much larger scale. Another advantage of these low-cost sensors is that they're full spectrum. And full spectrum obviously has a lot of advantages. The sounds are captured at a high sampling rate and are saved to an internal memory card. And they can be used for continuous surveys in real time, so they continue recording all bat species that are flying around at that time during the night. And then the calls that are recorded, we can use software to look at the sonograms for sound analysis. And so the sonogram is a graphical representation of the call. And we get a lot more information with full spectrum calls, clear call shapes, good structure, so ideal for sound analysis. So to summarize the approaches taken by BCT in developing our PAM surveys for the National Bat Monitoring Programme. So obviously we developed a new survey for NBMP called the British Bat Survey. And this really much used latest development in acoustic sensor design, automated call recognition, and interactive volunteer feedback. Various aspects of this sort of survey protocol was piloted in 2017 and 2018, and we also focused on trialing the AudioMoth to assess its usability, sensitivity, and recording quality. As we aim to launch the British Bat Survey nationally, we have been developing, obviously, an automated end-to-end system for monitoring bats that include low-cost full-spectrum bat detector, an app that will allow volunteers to send audio recordings to central server for processing, and a suite of software algorithms that automatically detect and classify bat calls to species, and an online portal that will provide sort of like a rapid and engaging feedback to volunteers taking part on our PAM survey. So there's a work that we've been working on, the sort of various things that we've been developing with our PAM work at BCT. So I'm going to talk about a pilot that we carried out in 2019 when BCT collaborated with Forestry England to undertake a groundbreaking pilot in southwest England to explore the ability of the emerging technology and the IT acoustic system that we've been working on to inform natural capital counting and woodland condition monitoring. And why was this pilot so important? Well, it's recognized that certain groups of species can be used to indicate the status of biodiversity as a whole, and also be used to provide an indicator of woodland health. In recognition of this, indices for butterflies and birds have been developed with environmental NGOs and are currently included within the natural capital counting for organisations like Forestry England, which are England's largest land managers. And this brought up the question of what other species could be included. And obviously, and bats were brought up. And why not? Bats can be an important indicator of woodland biodiversity and condition. All 17 UK breeding bat species obviously make use of woodlands and woodland habitats, whether for foraging, roosting, or commuting. UK bats are insectivorous, and the invertebrate community associated with woodlands and woodland habitats are a key food source. They are long-lived species. They interact with the environment both at local and landscape scales. And they're a vital part of our native wildlife. They account for almost a third of mammal species in the UK, and they occupy a range of habitats from wetlands, woodlands, woodland pasture, open land, farmland to urban areas. And they can tell us a lot about the state of the environment because they're top predators of common nocturnal species. They're very sensitive to pressures that can affect a wide range of taxa. So that includes obviously landscape change, agricultural practices, habitat management, and infrastructure development. And as mentioned before, they are all reliant on woodland and woodland habitats in a greater or lesser degree. So for the Forestry England 2019 survey, we used AudioMoth sensors to record the bat echolocation calls in the field. We surveyed 16 woodlands in total, 60 units were used. We needed to deploy these AudioMoths in protective casing because obviously when you use them, they don't come with an external sort of case. So, and it's needed to protect the microphone and the circuitry from the elements. So we tested an alternative case that was made from a modified waterproof push-fit junction box that you can see in the slide here that was designed and produced by Adrian Bicker. And having sourced our equipment and decided on our study site and methodology, we then used the pilot to test the acoustic monitoring IT system that we've been developing for our PAM surveys. As you can see from the slide, the basic premises is that a volunteer deploys a remote bat detector, which then records the bat calls. The recordings are then uploaded to a server, which are then uploaded up to a cloud storage, where then artificial intelligence algorithms developed by UCL and University of Edinburgh are used to detect and ID the bat calls in the recordings. And then the volunteers receive a report of what they have found. And this was really successful with them through this pilot and worked really well. And the results were really quite impressive. 400 surveys were carried out that took place over 36 nights across 16 woodlands and 60 monitoring locations. And it generated 9.5 terabytes worth of data. 45,683 audio files were recorded, and after classification and post-classification rules were applied, contained just under 2 million calls. And 8 species and 2 species groups were detected across the study area. Now, the 2019 Forestry England survey was designed to contribute towards Forestry England natural capital accounts, as we were able to provide a measure of species richness, species evenness, and an index of species activity for each monitored site. This meant that if repeated annually, this survey will also provide trends in species occupancy and activity to inform natural capital accounting and woodland condition monitoring. And long-term, obviously, the data has a wide range of potential applications, including research, change management, and obviously monitoring progress towards commitments in our environmental targets. Obviously, there's still a lot to be looked into this, and we need further pilots to go and test various aspects of the sort of protocol. But it's a good start, and we're very happy with where we've got with this pilot so far. So what's the development and future directions at BCT with our PAM work? So since 2019, our work at BCT with passive acoustic monitoring has broadened out to several other surveys and work areas. The work with Forestry England demonstrates just one of the ways that we plan to use the acoustic monitoring system. The system is now a central feature of an integrated model that includes bat monitoring, call verification, data analysis, and guidance. And we're now working with various landowners to implement this model on their land to help them monitor bats, assess ecosystem health, and improve land management. So in terms of British Bat Survey, because of COVID-19 restrictions, we decided not to run any further pilot of the British Bat Survey last year, and we won't be running it this year. Instead, we're going to focus our field work on testing the best way to deploy passive acoustic sensors to maximize their effectiveness. So far, we've investigated the impact of weatherproofing, deployment height, but also the angle and the use of amplitude and frequency thresholds on sensor sensitivity. So these are new firmwares that Open Acoustic Devices has recently released for the AudioMoth. So we're testing them out. And obviously these findings are going to feed into the development of a whole suite of new passive acoustic surveys. One such survey is the Night Watch survey, which is going to be launched this year. It has a dual aims of collecting robust, locally focused biodiversity data, while at the same time engaging a wide range of people from diverse backgrounds and abilities. Now, initially for this year, we're going to be targeting urban areas, but our aspiration is that the survey will become multi-taxa, allowing us to work in partnership with other species NGOs to collect robust monitoring information for a range of nocturnal species. It will follow a similar protocol used for the Forestry England survey, and obviously we'll be using AudioMoth acoustic sensors. In terms of woodland monitoring, building on success of Forestry England pilot study in 2019, we're now working with Forest Research to integrate passive acoustic monitoring of bats into the National Forest Inventory Field Survey, with the aim of undertaking field studies this summer. And we're also supporting Bat Conservation Ireland with their woodland monitoring in the Republic of Ireland. For the Ancients of the Future project, as we had many sites to monitor, they were quite spread out. And the volunteers we had very little or no experience of bat monitoring. We decided to utilize this passive acoustic monitoring protocol and this was carried out during 2019 and 2020. 2018, 2019. We used a similar survey methodology and recording schedule to the British Bat Survey and Forestry England Bat Survey. And for our target species, the noctule and the barbastille, the ability to deploy, a number of AudioMoths on some of our key sites meant that we were able to get a snapshot of activity. And this meant we were able to provide species distribution for each site, some habitat management recommendations, and bat activity maps that can be used to identify hotspots of activity for future monitoring or advanced surveys such as trapping to collect information about condition and breeding status of bats. Now, we will, of course, be providing landowners with all the acoustic results, but with the caveat that not all our species calls are manually checked, as our focus was to check our target species. So just to show you some examples, focusing, of course, on our noctule and barbastille. So for Breeden Hill National Nature Reserve, you can see where we detected barbastille and noctule calls from 3 separate surveys that were carried out once a month during July, August, and September. And then obviously the map below shows the activity map where we detected the most activity during one night of acoustic monitoring in July. Again, same with Dixton Wood. But we had 4 survey nights carried out once a month from June to September. And again, the map below shows you the activity that detected at each survey location. And again, same with land hydrock. So by using AudioMoth to survey for bats, we're able to give landowners an idea about the bat species using their sites, but also it means that they can then consider bats in their future management. like enhancing linear features, improving connectivity to wider landscape, consider minimum intervention, continuity of habitats and the right tree species, as mentioned by Alice earlier on, and retaining standing deadwood and trees supporting potential roost features. Also, the data can feed into future monitoring plans like more PAM surveys or transect surveys. And in addition, the activity maps will provide possible locations for trapping so that advanced bat surveys like radio tracking can be carried out to locate bat roost, tree roosts by experienced bat workers if that is an objective of the landowner and the site. So yeah, that was a quick overview of the work that we've been doing on passive acoustic monitoring and obviously using AudioMoth. So thank you very much for listening.
Many thanks, Sophie. That was superb. I love hearing about all these emerging technologies because I just get such a game changer in, understanding the wildlife we have here. So next up, we've got Vikki Bengtsson, who's the senior ecologist at Natura in Sweden. Vikki's going to be talking to us today about veteranisation and does it work for biodiversity? So Vikki, over to you.
And I'm also chair of the Ancient Tree Forum. So old trees are very close to my heart. I've been involved in it as an organisation for almost 30 years. So I'm going to give you a whistle-stop tour about some work I've been doing in relation to veterinisation and using tools instead of time. And hopefully you will have listened to Carl and Izzy earlier this morning and got a bit of the background to what this is about. But just for those of you who weren't there this morning, it's a nature conservation tool which is about creating decaying wood habitats on live young trees and trying to mimic what happens in nature using tools, but never on veteran trees. And it's about bridging the gap, which I think Carl and Izzy talked about beautifully. This photograph on the right here is a hornet that's looking out at me from a chainsaw created woodpecker hole. So just to give you a very brief background, I've been working with veterinisation for almost 30 years and started thinking about it in relation to restoration of wood pasture. So working on clearing around old trees where we had secondary woodland that's grown up. And it just always felt difficult to remove some of those trees that are kind of getting up towards sort of early maturity when they're not really interesting in terms of habitat point of view and all the trees we're talking about over the course of today. But it felt almost like they were a resource. So there was a kind of discussion that we had, and not just me, but others too, about, is there a way in which we can damage the trees so they're no longer competing as strongly, but that they still stay alive and start to develop some of these kind of features that we so value in our old trees, not to replace them, but to kind of help bridge the gap. We've all got this issue of generation gap, and it's an issue across all of Europe. I currently work in Sweden quite a lot, and we have the same problems there. And the issue also of overgrowing around our old trees is also one of the most common problems our old trees face in terms of their future survival. So we started off a bit of, I guess, back of a fag packet, really, using climbing spikes, topping the trees, using a sledgehammer, doing kind of hard topping, really, rather than pollarding. And we even had a go with explosives, which was a lot of fun and a crowd pleaser, but not sure it's really the technique that I would be suggesting for going forward. But what is interesting is some of the trees that we started working on at Hatfield Forest, where I used to work, which is one of the National Trust sites with lots of old trees, is that we're seeing cavities forming. So we're seeing decay happening from work that we did in the late 1990s. However, the big problem there was that we didn't really have a good monitoring system in place. We did very detailed records of all the trees that we did back at Hatfield Forest at that time, but unfortunately this is pre-digital, and that you can tell by the grey hair, and that all of our records were lost in a flood, which absolutely devastated me. But these things happen. So the big question, does veterinisation work, wasn't quite as easy to answer from the work that we'd done at Hatfield Forest. And I happened to take a group of Swedish nature conservationists to Hatfield Forest, and we got talking in the pub, as you do, after having been out all day, about veterinisation, about monitoring it, about understanding whether or not this is actually a valuable tool to add to our nature conservation toolbox. And the more I started to look into the research, the limited amount of research there was out there about this. Now, there's a fabulous piece of work from North America talking about bears being ecological engineers. And this photograph was taken by one of my neighbours in the north of Sweden, showing a bear, a young bear, climbing up a pine tree. And you can see that the bark is flying in all directions. And I could see this is actually the same story happening in Europe that they were writing about in America. So just a bit of background to that pub discussion about, right, okay, well, veterinisation is promising, but we need to get some good data in order for it to be financially worthwhile and for us to spend those limited resources we have. So I'm going to give you a whistle-stop tour through a project that I've been involved in since 2010 when the first idea arrived. And we set up this international trial. It started off in Sweden, but we also have 3 sites in England and a site in Norway. I'm from Scotland, not from Sweden. So for me, it was important to have some sites in the UK too. And we focused on oak. And the reason for that is, as Ross and others have already mentioned, oak is one of the species that hosts the greatest number of saprophytic species. And I include fungi in that, as well as the invertebrates and lichens and others. So it's a hugely valuable species, and only really is equivalent of pine in terms of its long-livedness in our climate, at least. So this project, we managed to get 20 sites, and at each site we have 49 trees. Now it sounds a bit odd having 49 trees, but it's 7 by 7. So we have, all the trees are healthy, 25 to 60 centimeters in diameter. They're all tagged on these sites, and they're basically quite boring oak trees, I would say, 60 to 100 years old. And we tested 5 different treatments, veterinisation treatments. So these are mechanisms that we wanted to have both short, quick fix, if you like, and ones that would take longer to show the results. So we've got one that we call a horse damage, and Carl mentioned that they were doing that at Sherwood. This is a broken branch, and again, many of the cavities that form are often in association with either dead branches or broken storm-damaged branches. We've also ring-barked large branches. Then we created 2 cavities, one which was to look a bit like a woodpecker hole, and the wood was carved out into the heartwood, which is a relatively small hole, but the entrance was kind of 12 by 8 centimeters. And the one on the right is what we call our kind of nest box version, but it wasn't really to be a nest box for birds. It's about creating heartwood decay in the tree. We also topped the trees too with a natural fracture cut in order that these trees wouldn't fail. So those are the 5 treatments. At each of our sites, we have 7 trees which had one of each of these 5 treatments, and then we had 14 controls. And that's one of the interesting and the strengths of this project. It wasn't set up as a university study, in fact. We did it really with financing from all of the landowners. So it's really exciting and shows a fantastic way in which we can kind of all work together to get a big piece of work done that will help us with practical nature conservation. So then, does it work? That's the million-dollar question. So there are 3 different things I'm going to talk about here. One piece of monitoring we did was in 2014. That's only 2 years after the work was done. We put up window traps on control trees and on the woodpecker holes and the nest boxes. And we collected and we identified the beetles, the Hymenoptera, and the flies. So those 3 groups. We have only just got round to analysing that data. It took several years to get finances for the identification. So that's one of the problems with these kind of big projects where you don't have long-term financing. But it's been really exciting, and more exciting than I could have imagined. We got more than 500 species of Coleoptera from the traps, and almost double the numbers of individuals and taxa in the nest box and the controls. And in fact, we had more than the controls in the woodpecker hole. We had the same pattern for all the data, and that's what's really exciting. And we also are looking, we're doing a bit more analysis there, looking at some kind of more interesting species, if you like, more closely, things like Lamexilon navale, which we know likes the kind of exposed sapwood on trees. And the other thing that's very interesting is that we divvied up the Coleoptera in particular into the kind of guilds. So the directly wood-living species, the indirect wood-living species, fungi-associated species, and others. In other words, the species that are not really interested in trees. And that's also really interesting because there was no statistical difference between the controls and the cavity trees that we've created, in contrast to the other guilds. Now, this work hasn't yet been published, and I hope that we will publish it. So for flies, the bees and the wasps, we didn't have quite enough data with regards to the Hymenoptera, but we did have hornets, and we did have this fabulous yellow-ringed comb horn crane fly, which became a bit of a kind of mascot for the project because we found it as well feeding on the sap runs associated with the climbing spikes. But again, more than 500 species of fly, and looking at least an overview, we've got lots of species associated with sap runs particularly, and wet decaying wood and rot holes. So again, promising results there. So then the other piece of work that we did was to look at fungi, because from my point of view, it was the decay fungi that were really interesting in this project. We want the decay fungi to get going to create the conditions for all the other species that come later. And in 2018, the research group from the Agricultural University in Sweden and Norway got some money to take samples, and we did DNA analysis. And there's a paper published on this, so if you want more information, that's available. And it's open access too. So, but what you can see with this scatter diagram here is that we do have different species compositions for the control trees compared with the nest box and the woodpecker hole compared with the horse damage, which is what you might expect because you've got sapwood exposed here, nothing happening with the controls, and heartwood and sapwood exposure for those treatments. But what is fascinating is that we found about 300 different species, primarily endophytes, ascomycetes out of this data. So there was this whole kind of hidden life of oak trees, and I'm sure that Matt will mention something of this as well. And this is information that other people are finding with the DNA analysis. But what was also interesting is we had kind of double the quantity of DNA in the treated trees than the control trees. But we perhaps didn't have quite the number of wood decay species that we might have liked. And these 4 species, Laetoporus is a great one, and Fistulina was only from 1 tree, but we found a rare species in Sweden at least, Inoculation stryophylla, which was in 2 of our trees. But again, it was less than any of us had hoped. And I guess what that suggests is these things take time. And that's no surprise. Those of us working with old trees that live 500-600 years, tree time is a completely different concept. So moving on, last summer we revisited 18 of the 20 sites. COVID did give me some problems, as others have mentioned already. This is just an example of a fracture cut with a lesser spotted woodpecker drumming like mad on that fracture cut. So this is after 8 years. And the results from that work, we saw that the nest box had the biggest impact on the growth of the tree. And again, that's not surprising. We took both the top off and also took a big chunk of the central wood from that tree. The nest box and the woodpecker hole had more decay than the other treatments, and that's really encouraging. In fact, almost 40% had some kind of decay happening after 8 years. But it had limited impact on the tree elsewhere, and I think that was also interesting for me because the actual veteranization is really having an impact. So we're seeing that there's cavities forming, but there are no other naturally forming cavities in the control trees, for example, and there are no other naturally forming cavities developing at the moment on the trees that we've treated. Also interesting was that the woodpecker holes and the nest boxes that have calloused had lots of damage on the callus. Now that could be woodpeckers, but it could be something else. And that in itself is interesting, a whole kind of ecological story there that I don't have the answer to. Some of them had completely occluded, so the hole is no longer open, but that doesn't necessarily mean that's a problem. We would hope there's dysfunctional wood there, decay will happen, maybe in 20 years' time a woodpecker will open it up again. We had more sap runs associated with the nest box and the woodpecker hole, and that doesn't really surprise me because they use climbing spikes, and that kind of damage seems to be promising in that respect. Aspect didn't really show any impact. And also, I think what we could say was that doing treatments on the branches after 8 years, we didn't really see any great benefits. In fact, many of the oak trees or control trees had large dead branches of the same kind of size. So I think if you're going to do branch treatment, you've got to go a bit bigger. So you need to be looking at 20 centimetre size branches really to get, or ring barking the tops, which is the other thing that Reg has been doing at Sherwood. The other thing that was interesting was almost 70% of the nest boxes had been used for bird nesting. And I think, as we know, this is part of the kind of whole ecological process of cavities, that bird nesting material decays and breaks down. It's part of this kind of gungy wood mold stuff that we find in our very old trees. We did also find bats, live bats and droppings from bats, as well as insect remains from bats eating, but they're probably under-recorded. Very few trees died, and I think that was one of the kind of big questions for me, was the aim was to keep these trees alive, and very few have died over that course of time. In fact, more have died because they've been felled inadvertently. That's not included here, but some of the trees have been felled inadvertently. We also found wood mice, live wood mice and wood mice remains. And we found lots of tree ants, and that was really exciting for me because that's kind of showing us that ecologically these are similar to what we find in nature. So I just want to finish off by showing you a few kind of pictures. I'm hoping this is a really, really bad quality video, endoscope video footage of some brown long-eared that we found inside this nest box. This is another angry bat, very angry at me sticking my endoscope in, and that was inside one of these as well. And here we have another pipistrelle that's hanging on the roof of woodpecker hole. That's what the woodpecker looked like, and you can see the damage on the callus growth. You can also see the bird nesting material. So again, these cavities have got a kind of whole host of different species using them both in space and in time. This is one that I've got in my garden, and it's quite common that the tits are using them, but also starlings and nuthatches. Here's the wood mice that we found inside one of the woodpecker holes. Here's the damage that I've been talking about. So this is possibly woodpeckers, but that looks more like teeth marks. And we don't have grey squirrels in Sweden. So this may be something else going on there. I'm desperate to put some cameras up to see what's going on. Other things we found, this is from bees that have used the cavities and then in turn have been kind of used by moths. Here, there's fresh oak leaves that no one can really explain to me what's brought it there, but that was also well after the bird nesting season. We've got ground beetles using the sap runs. We've got these kind of lifting bark that we know is a potential bat roost feature that wasn't one of the target features. So just to finish up, this was a whistle-stop tour, and I'm sorry if it's gone really fast, but I could probably spend another hour and a half talking to you about the results. It's so exciting. But I think what it does say to me, at least, is that we're on the right track. We've got the tree ants have moved in. We're getting sap runs. We're getting species, different species, using the cavities in particular. And I think it's the cavities that really are where we have the lack of that feature in our landscape. But horse damage has given us some really good results in terms of decay. It's easy and cheap and quick to do as well. And I know that one of the discussions that I've been having over the years is we wanted to do inoculation when we started this work, but it was just too challenging to get all the permits we required to do it. So I'm really interested to hear what Matt's going to talk about following on kind of potential doing veterinisation in combination with inoculation of the wood decay species we're interested in. But of course, you know, it's never a replacement for our ancient trees, and they're so important for us to retain and look after that for all the money in the world, that's where we should be spending our money, looking after these fabulous old trees. But this is an extra tool for us in our toolbox, which might help bridge the gap in terms of both generations and habitat. Just a quick thank you to all those organisations that have both financed and supported the work. And also thank you for listening. This is a red squirrel sticking its head in the one in my garden on Christmas Day.
Thanks, Vikki. I mean, it always surprises me how quickly, you know, species will colonize new habitat, you know, because we assume that a lot of these things that are rare, a lot of these species associated with deadwood habitats have poor dispersal abilities, but I think a lot of them are actually very good at getting to new areas of habitat if they're there, if they're available. That was really interesting. Thanks very much for that. And so a natural follow-on from that talk from Vikki is we've now got Matt Wainhouse, who's an ecologist and a PhD student at Cardiff University. Matt's going to be talking about, can we make a veteran tree, artificial inoculation of heart rot in forest conservation. Can I also add, though, as well, if you've got any questions, please just put them down in the live Q&A straight away as you have them, and then we can see them here on the platform. You can do that in the platform on your screen. So as soon as you have a question, just put it straight down, and then we can see it. So Matt, if you'd like to take it away, please. Thanks very much.
Okay. Hi, everyone. Thanks, Ross. I'm going to sort of talk about some of the work we've been doing with Back from the Brink, looking at the conservation and the ecology of heart rot species. And some of that work is looking at how we develop inoculation of heart rot as a conservation tool. So I think we've laboured this point enough today. So I thought instead I'd actually just read a little bit of a poem by Robert Macfarlane. It's Heartwood, and it's in the book Lost Spells, but I think it kind of captures the point beautifully. I am a timekeeper, breath giver, deep thinker. I am a city of butterflies, a country of creatures, but my world takes years to grow and seconds to crash. And I think that really sums up what we're talking about here with ancient trees. They are these wonderful structures full of life, but they are quite threatened. And we've talked about this this generation gap as well, and we'll come on to that shortly. So we've got some good news, which is lots of tree planting. We've had recently, the most recent press release from George Eustace was on woodland creation. I find this sort of slightly irksome because can we really create woodland? Woodland creation takes time. Woodland it's a canopy, it's tall trees, it's shade tolerant flora, and it's a very unique set of fungi and insects. And of course, the bit that I'm interested in, lots of deadwood, and in those standing trees, cavities as well. And so the cavities that I'm interested in, I guess, it's all about the heart rot fungi. Now, for those who are less familiar about the fungi, heart rot are a group of fungi, and they're the ones that are able to get into the center of the tree, and they're the ones that are going to be doing the hollowing out. So they're specialists. They can get in there. They can cope with the metabolites within the tree, all those toxic compounds. They can deal with the low oxygen and start to decompose that lignin. But it's only a few that can do this. But as they start to decompose that lignin, it's providing, it's changing the environment to such an extent that beetles can start boring their way in, and other filamentous fungi can start making their way in, and we start to develop over a very long period of time a very biodiverse, very rich community. But like I say, this does take time. So we think that for a beech tree, for example, it's probably about 80 to 100 years before the heart rot fungi are able to even access the middle of the tree and start that decomposition process. Whereas in oak, it might be 200 years from some of the research that we're doing to the side of this work. And then the cavities themselves are full of lots of rare species and networks in their own right, like the sexy pin up there of the saproxylic world, the violet click beetle. And as in the same way we have lots of rare and vulnerable insects associated with heart rot, a lot of the heart rot species themselves are also very, very vulnerable. This is sometimes overlooked. A lot of them are adapted to old growth forest, to large volumes of wood, and so many of them are kind of stuck in places like Windsor or Moccas, unable to escape, even where habitats and woodlands are managed in a way that would be favourable. So we've got policies, and for these sticks in the ground, we've got policies for the ancient trees and our ancient woodlands. We've even got policies that protects these bits in the middle. But what we are lacking, and what we're trying to do, is create these tools that will help all of these old growth associated species move into some of these younger blocks of woodlands. If we can't do it for bridge this 200-year gap, we're never going to do the 500-year gap. And so one of these tools is heart rot inoculation, and that's a bit that we've been working on in Cardiff. And I feel a little bit fraudulent, I guess, talking about heart rot inoculation in a session about new tools and innovation, because I don't think it's particularly innovative. It's certainly not a new tool. The mustachiotoed hipster you can see in the picture there is Robert Hartig, one of the fathers of modern mycology, and he was inoculating trees with fungi 140 years ago. So there is a little bit of a precedent in what we're doing. It's mostly been around understanding colonization of timber in wood production, but in the 70s in the US they started looking at inoculation in terms of its conservation value, but really focused on woodpeckers. So creating heart rot in trees so that woodpeckers, so it's nicely starting to decompose, and the woodpecker can bore that out without getting a headache. And of course, the most American thing to do would be to load your inoculum into a gun and blast it into the tree. And so that was their protocol. The process itself is deceptively simple, perhaps. It's you take your fungus, you isolate that fungus, you culture it on your inoculum. You take your inoculum out to the woods, you cut a hole in your tree, shove it in, and then, as with any good conservation intervention, you do the monitoring afterwards to see whether it's working. Now, the devil is in the detail. So yes, a simple process, but actually coming up with a protocol is perhaps a little bit more challenging. Which fungus are you going to use? Are you choosing a fungus that's associated with a specific beetle? Is it going to be a white rot or a brown rot? Is the rate of decay important? Is the fact that it's dispersal limited important if it's a translocation? Is how many strains you're going to use? Which tree species you're going to inoculate in? How does that tie in with the fungus you want to use? How many trees are you going to inoculate? Where on the tree you're going to inoculate? And how is that going to affect where the cavity and the decomposition forms beyond that? So there's lots of questions that feed into the design. And then just to step back a moment, I guess, is to think about when is it appropriate to inoculate. And we've kind of identified two scenarios. And Vikki touched on it just then, is the first being that inoculation can be used really as an extension of your veteranizing methods. So whereas veteranizing, deliberately damaging a tree like this car in the bottom, is dependent on the random arrival, the stochastic arrival of the fungi in the tree. What we're doing with inoculation is exactly the same, but we're using a specific fungi to elicit a very specific response. The second situation that we've got where we think this is going to be a useful technique is in translocation and the reintroduction of rare species. So I mentioned some of these old growth heart rot fungi that are very isolated in the woodlands that they're in, helping them to with assisted colonization of new woodland. And so we've started 2 trials with looking into both of these scenarios. The first of these is a veteranizing inoculation. So here we're inoculating young beech much in the same way as we would in the veteranizing oaks that Vikki was talking about. We're doing this in younger trees than they would typically get heart rot. And we're cutting the holes in to the trees. We're using rather large wood blocks, about 10 centimeters cubed, pushing these into the trees and compacting them with inoculated sawdust. And we're using 4 fungi in this, 2 that we think are quite important for some of the commoner, more aggressive species that are hollowing out beech trees, and 2 that are a little bit rarer, but very specific to Windsor. Now the second trial that we're running is for the reintroduction of 2 very vulnerable species. So these 2 beautiful things, the one on the bottom there, the lion's mane, Hericium erinaceus, and the one on the top is Hericium coralloides, the coral tooth. And both of these 2 have gone extinct in our trial woodland. There's records going back of both of these species within the last 100 years, but neither have been seen for a very long time. So they may be under the radar, but we believe they're extinct. So we're reintroducing them. We're using 2 strains of each. The strains have been sourced from the south of England, as close as we can to the site, and we're trialing 2 different substrates, solid timber and a sawdust and grain mix. But these take time, and so once they're in the tree, it really is a waiting game. Now, one of the issues of some of the previous studies has been a failure to sort of ensure this long-term monitoring. So we don't really know how these are functioning, these inoculations are functioning over the long period, over their development. And so we're putting protocols in place to do this, and the first of these is through fruit body surveys, and all of our, in the short term at least, all of our species bar 1 over both trials have fruited which is very exciting, including this tiny little lion's mane on the end, this Hericium, which we hope was spreading spores around this woodland for the first time since it went extinct. But although this tells us that the tree is alive, the fungi is alive, sorry, what we don't know is whether it's actually established within the tree. And so for that, we're using a couple of different techniques. The first of these is sonic tomography. So this is looking at the density of the wood. And by repeating this on some of our inoculated trees, we can see how the density is changing over time. So we've done this in 2018, 2019, and hopefully again this year, and then way off into the future, we hope. But even just after 1 year, we can see that we've got these purple and these green blobs of color, which mean that something is happening within the tree, that there's been a change of density. Now, there are several reasons that might have occurred. It might be the decomposition from the fungi itself, but could be dysfunction within some of the tissue. It could be compensatory growth. We don't know. So we've really got to watch and see what goes on and how that develops. So the third monitoring method that we're using is to take samples from the tree itself. So we're doing this in 2 ways. One with a drill, like you see Ian doing there, and the other with an increment bore and extracting a long core of wood. We bring these back to the lab, we put them on agar, and we see if our inoculated fungi grows out of them. If it doesn't, then and that's often the case, because we often find, that we have lots of interlopers, so yeasts, bacterias, other filamentous fungi, who are just too competitive to allow our fungus to escape onto the agar. And so what we have to do is look at the DNA within the wood. So using DNA barcoding, grinding up our wood samples and probing them for the DNA, and we've had some success with this so far. So I guess this whistle-stop tour is I guess coming to an end now, but do I think it's going to be a useful conservation tool? Well, absolutely, yes. I mean, within the scope of this project, we've been able to see that we can get fungi established within the tree. Whether these are going to become fully functioning cavities, we probably need another 50 years to find out. And whether we're going to have woodlands packed full of lion's mane and coral tooth in 50 years' time, again, we need to carry on that monitoring to really find out how it's going to work. But for some of our old growth fungi, for these important heart rot species, the only really method that we have for their conservation to allow them to colonize new sites is through this method, we think, through inoculation. Whereas for veteranizing inoculations, we've seen that there are other techniques, and like the veteranizing, like the invert boxes that Steph's going to talk about shortly. And so this really is probably a tool to sit alongside those. And so we can have a hybrid approach really that helps our ancients into the future. So I'm going to leave it there with a massive thank you to the funders on the right and the superhumans on the left. Contact details there if you have any questions. So thank you.
Thanks, Matt. Really nice proof of concept study there, and really good to see you got some success from your work so far. So next, leading on from that, we've got Steph Skipp. Steph's going to be talking about, Steph's actually a PhD student at the University of East London, and she's going to be talking about whether beetle boxes help deadwood insects cross tree age gaps. Now, hopefully, she might let us see some of her squishy beetles that she's so fond of. If you don't already, then please follow Steph on Twitter, where she posts lots of really cool pictures of dissections of some tiny beetles that are smaller than a full stop. She's very skilled in that. Okay, Steph, over to you.
Great, thank you very much, Ross, for that introduction. Yeah, do go and check out my tiny minuscule beetles. I'm going to be showing you a few of those on this presentation. But yes, I'm a PhD candidate from the University of East London, and I'm going to be talking a bit about the beetle box project that I'm doing as part of the Back from the Brink project. So kind of back to the beginning, I'm sure we've all covered this quite a lot, but just talking about that little term saproxylic. I've been doing this PhD for 2.5 years and I'm still not 100% how you say it. Lots of people say it differently. But yeah, saproxylic describes an invertebrate that requires deadwood to complete its life cycle. So that can be in a range of ways. Some invertebrates spend their whole life amongst deadwood. Some eat deadwood, some eat other invertebrates that live on deadwood, and some of them just use deadwood for a certain part of their life cycle. So you can see this little click beetle spending its vulnerable pupal stage protected underneath the bark of a tree. So you can see that there's a whole wide range of different ways that invertebrates can use deadwood, and that leads into the massive diversity of invertebrates that occur in deadwood. So another reason why deadwood is so diverse is the fact that there are so many different microhabitats associated with trees. We've seen a little bit of this already in some of the previous talks, but this image shows you some of the microhabitats you might find. So there's a bit of peeling bark. You get some specialised flat beetles that live underneath that. You get some saprons, so that's lovely nutrients for some invertebrates. And then you get these big old hollows. And I'm going to be talking about when these hollows occur at the base of a tree. But inside these hollows, you do get some lovely, nutritious wood mould that invertebrates just really love to live amongst. So because of all those different features, there's a massive diversity of these invertebrates, as I said. So there's about 1,800 species of invertebrate and around about 700 species of beetle. And you might think like, oh, these invertebrates, there's lots of them, but they're all tucked away inside these trees. Why do we have to worry about them? But it is really important that we conserve these invertebrates because they all contribute different functions to woodland health. So there's been a recent study by Stephen Falk talking about how some of these invertebrates contribute to pollination. So they visit flowers. A lot of invertebrates contribute to the recycling of nutrients in woodlands. So they help break down wood and return the nutrients into the forest floor where it can continue to nourish the woodland. And also you get some predators and parasitoids. So you can see in the center here, there's a wasp, and I think that's one of the ichneumons. And they are parasites on certain other species. And so the presence of these parasites and predators help to control some species that could otherwise have population explosions and become pests and start causing some problems in our woodlands. So it just shows how important it is that we look after all of these different species. But as we've heard so far, there are a number of threats to the species, and a lot of them involve a lack of habitat. So a lack of these trees that have these decay-related microhabitats that the invertebrates can use. And a reason for this is because, well, one of the main reasons is that in the 1700s and 1800s, there was a massive felling of lots of trees to use for naval ships. And so that contributed to trees of one age group almost completely being wiped out in lots of areas. So that means that as the current really old trees start to decay beyond usability by these invertebrates, we aren't having trees developing these microhabitats, allowing invertebrates to persist in the landscape. So that's not ideal, and we need to think about how we can fix this before we get some extinctions. So I know you've seen this picture a few times, the best picture on the internet of the violet click beetle. I'm going to use that to introduce the beetle box concept. So aside from veterinisation practices that we've heard about over the last 2 talks, I'm looking into a potential man-made solution to the problem. So the beetle box is something that we can introduce in woodlands to artificially introduce a little bit of extra wood mould habitat. So you can see here the box on the right looks a bit like one of the cavities that you might find at the base of a tree, and that's the specific habitat that the violet click beetle loves to be in. So it's kind of this intermediate transitionary habitat between soil and decaying wood. So that's what we're trying to replicate, and as the violet click beetle is an umbrella species, so it uses just the best of the habitat, we know that if we were to find one of these beetles in our box, we would be creating this habitat perfectly for all the other species that might use it. So you can see on the left, this is what we're aiming for, some lovely crumbly wood mold. It looks just like soil, but there's a lot of nutrients and a lot of really good stuff in there that the invertebrates are going to love. And you can see on the right, this is what our beetle box looks like before we've installed it. So we partially bury the beetle boxes into the ground. There is a below ground aspect of it, which helps us to achieve this intermediate sort of conditions between the soil and decaying wood. The beetle boxes were inspired by some previous studies. There's a research team in Sweden that includes Niklas Jansson, and he's really a driving force behind lots of the beetle box work. Previous beetle boxes looked a bit like bird boxes that you might have up in a tree, and that's because, as you can remember from my image that I showed you, that you often get these hollows aerially up high in the trees. And so that's what he was trying to mimic with these beetle boxes, and he had quite a lot of success with that. And this on the right is, I think, one of his most recent endeavours in orchards. So yeah, he's trying to provide lots of habitat for threatened beetles. But so this is my project is a bit of an extension for that, but we are still actually working with Niklas to investigate basal hollows, so the ones that are down in the ground. And here, actually, we have Niklas here on the right of the screen as he came over to help us with our beetle box installation. And this was one of our installation days. Got to say thank you to my supervisors, Caroline and Stu and Jack. And there's also Sam from the SRA who helped me to install these boxes. And then also my family came in and helped us to fill them up with all the lovely ingredients for the beetle boxes. But yeah, so we've installed these boxes, and there are also some beetle boxes installed in Sweden. So potentially later on in the beetle box life, they can have some comparative studies between here and Sweden. So where we put the beetle boxes we were led by where the violet click beetle is. So we've got Bredon Hill and Windsor Forest. So those are 2 of the 3 locations that violet click beetles have been found. There are slightly fewer beetle boxes installed in Bredon Hill. That's just because Bredon Hill has got some slightly more difficult conditions for installation, because it is actually, as the name suggests, a hill. So yeah, it was more difficult to find sites for the boxes. So there are fewer there, and we're trialing a few more different types of box in Windsor Forest. So the types of boxes that we trialed. So you got what you might think of the sort of classic beetle box. You would obviously think to make your beetle box out of wood, as it is mimicking a tree. But one of the issues that has been found in previous beetle box studies is that wood can slowly warp and change over time, and that can be a bit of an issue in terms of the longevity of the beetle box, because as it's mimicking a tree hollow, those can persist for hundreds of years, and you're not going to quite achieve that with a wooden box. So we also decided to have a look at some other materials. So yes, that one in the middle is a wheelie bin. We did some extensive investigations into things that we could use to create a more durable plastic box, and the wheelie bin just met all our all our requirements. So yes, that's an adapted wheelie bin beetle box. And then we've also more recently created a concrete box, along with the help of the some of the students at the University of East London. We created that a little bit later than the others. As you can tell, it's a lot of work to create a wonderful custom concrete beetle box. So that isn't included in the main trials, but we are using it for an interesting side trial as to what happens when you put beetle boxes in someone's garden. So we've installed that in John Little's garden in Essex, and yeah, we're going to find out what beetles that he has that might like to live in a beetle box. But back to the main study, in terms of Windsor Forest and Bredon Hill, I'm looking at a few different variations on the contents of the box. So most of the boxes contain oak sawdust, and that's because oak is really the easiest to get a hold of, so that's why. But in Windsor Forest is actually a beech woodland, and so we are trialing a few beetle boxes that we managed to get some beech sawdust to put inside, just to see if using the native type of sawdust makes a difference. But the hypothesis is that it shouldn't really make a massive difference, as we're aiming for the really decayed down stuff. And as wood decays away, you lose a lot of those specific defense mechanisms that certain woods have. So it does homogenize, and the sort of species groups that live inside it can often live on a variety of different types of wood. So we don't think there'll be a massive difference, but early on in the project, you never know. And then on the right here is a picture of some of the organic poultry manure that we've put in quite a lot of the boxes. We use this because, as has been said before, tree hollows are often used by birds who have to release their feces somewhere and it goes into the tree hollow, and beetles, we think, just love this. They love a bit of extra nitrogen to keep them going. So we're investigating how adding a little bit of fertilizer can affect that. So the project is quite far in now in terms of the PhD involvement. So I installed the boxes in 2019 and left it for a year to get all ripe and tasty for the beetles, the wood mold, to decay down into what we want. So last year, despite COVID, I did manage to get a few trips out into the field to do some sampling. So how I did this was I took out some samples of a fixed volume from the boxes and I sorted through them on hand. This is just in case we encountered our friend the violet click beetle. We wanted to make sure that we weren't damaging any populations, so I made sure I did. Spoilers alert, I didn't find any adult violet click beetles this time, but yeah, and it also helped me to collect some of the larger species that we found inside, so we did some sifting through. And I also took some samples from some of the trees on site with all the appropriate permissions, so that I could compare the sort of species that we're finding in the beetle boxes to those that you might find in an actual tree. Then after doing this, I took my samples home and I put them inside Winkler extraction traps. So these were a little bit of a panic invention. I mean, they do exist, but I was previously hoping to use Tolgren traps, which use a light bulb to dry out the sample. And as the sample dries, invertebrates will run down and fall into a collecting pot. But with COVID, I couldn't access the lab. I couldn't access the appropriate space to do this in. So I panicked, got loads of pillowcases, and I sewed them into these handmade Winkler traps, which work on a similar principle, but they just use kind of the air flow to dry them out over a longer period of time. But actually, this has turned out to be working really pretty well. You can see on the right, this is some of the beetles that you might get out of that trap. And actually, the majority of the species that I found were as a result of these traps. So yeah, I would definitely recommend these to the Sherwood, Carl and Izzy. I definitely think that this is something to think about. So yeah, I did find quite a good diversity of beetles inside these boxes, a total so far of 36 species, and I've only sorted through the samples from my first sampling session. I actually did 2, so that number's only going to get bigger. But yeah, we got a range of these species, some with lots of different requirements. So a lot of them like fungi, some of them, yeah, there's just a whole range of different requirements that these beetles needed. So it's really interesting to see, and it just shows that the violet click beetle is umbrellaing a whole range of really interesting fauna that you can find. But in addition to the beetles, I'm also looking out for things like pseudoscorpions, worms, myriapods and isopods, just to widen out the scope of the study a little bit bigger. But onto what we really want to hear about, which is what's going on so far. So with the first set of beetle box studies looked through, one of the interesting things is that there seems to be quite a strong difference between the beetle boxes that had the fertilizer added and the beetle boxes that didn't. So this is Breeden Hill, the average number of beetle species per box, and this is for Breeden Hill, the average number of individuals of beetles per box. So both of those, there seems to be quite a strong difference emerging, that beetles do really like a bit of fertilizer in their boxes. But interestingly, the boxes without fertilizer, we seem to be finding a whole different suite of species inside. So there seems to be a different assemblage that likes the additional fertilizer, and a different that doesn't like this additional fertilizer. So yeah, some really interesting things coming out of that, and I'll be going into that a little bit more detail in my thesis that comes out, will hopefully come out early next year. Onto Windsor Forest. So these are some of the trials that we did. You can see there's quite a lot more because of the increased number of boxes we put into Windsor. But similarly, there seems to be a difference emerging in terms of the fertilizer. So the wooden box that had oak sawdust with fertilizer had a lot more species than the wooden box that didn't have fertilizer inside. The plastic boxes, it's been quite interesting in that for a while it didn't look like the plastic boxes were doing as good as the wooden boxes. But recently we had one that just had loads of beetles inside it. And I think that has something to do with the moisture levels. I think that's something that really needs to get looked into, is the optimum moisture that certain beetles need. Because obviously the plastic excludes some of the rain and it doesn't warp, so it doesn't let in as much of the water. It's a lot drier often in the plastic boxes. So yeah, that's something we've been finding. Similar, also beech, the sawdust that has the that matches the forest type seems to be doing really well. That's kind of to be expected in terms of the beta boxes have only been out for a year, so the sawdust it's decayed quite a lot, but it hasn't decayed as much as we really want it to, like really ripe. Okay, so this is the next. This is a similar, just for the number of individuals of beetles, so you can see kind of similar things going on. But yeah, you'll definitely hear more about that in my thesis that comes out. And yeah, so it is really interesting that different types of beta boxes are producing different results. And in terms of advice to people, I'd say just try a range of different things. Fertilizer will guarantee you some extra beetles, but it can also be interesting to investigate what happens without it. So yeah, thank you to everyone who's made this project possible, including all the funders and my supervisors and other volunteers that have helped me out. Thank you for listening, and I hope to hear some questions later. Thank you.
Many thanks, Steph. That was really good. It's nice to hear there's another use for chicken manure, and also you've been getting lots of species over a relatively short period of time. Really nice to hear. So now we're going to have a Q&A session. So I think all the speakers are here with me to answer any questions. First up, I can see one here for Matt from Jane Sears. I don't know if you can see this, Matt, but it says, do you have any idea how long it would take heart rot to develop in Scots pine? This is in connection with our work to conserve the critically endangered pine hoverfly that depends on that habitat.
Oh, can you hear me?
Yeah, can hear you. Yeah.
Okay, sorry. So short answer, I've no idea. But equally, the heartwood of Scots pine is full of lots of very interesting chemistry, lots of metabolites in there, which are pretty toxic to a lot of fungi. So how long would it take for the heart rot to develop there? I mean, it's a long-lived tree, so I would guess it's a long time again, more comparable to oak than beech perhaps, but no, I don't know. I would certainly advocate giving this a go though. There's really nothing to lose from trying.
Can I just add into that there's some researchers in Sweden looking at veteranizing of pine, just because it is such a long-lived tree along the same lines as for oak. But what they're building on is the kind of traditional forestry technology where you kind of damaged pine in order to make the wood better and last longer. So I think there's some real challenges there to get the kind of decay processes going, because damaging pine kind of stimulates the production of much of their defense mechanisms, and that might make them less able to decay, or rather the fungi less able to decay them. But it's a project that's just started, so I'm watching that with.
Sorry, I'll add one thing quickly as well. It has been done before on various pine species, mostly in North America, in inoculation. I don't have those results. We're going to bring out a paper, so hopefully that'll the information will be in there whenever it gets accepted.
Okay, thanks, Matt. Thanks, Vikki. Vikki, here's a question for you. What do you think about veteranizing species other than oak? This relates back to the last question too.
Well, we've done some work on oak and beech in Sweden, and I think the focus has primarily been on the long-lived tree species, because that's where you've got the biggest generation gap. So the issue with pine is also interesting. But we have been doing some work on maple actually, and lime, where that's the kind of infill tree species in wood pasture restoration sites. And I think, you know, some people were talking earlier about horse chestnut, that, you know, I think that using the tree resource you have is important, but just consider the kind of cost benefit aspect, which I think is, you know, as a practitioner, that's always in my head, that some of these younger or the tree species that develop interesting habitats more quickly, it's probably less cost effective for us to work on those tree species. So, and ash obviously is kind of out the picture with regards to ash dieback as well, which would have been another natural species. And we actually did some ash at Hatfield Forest back in the 1990s. So yeah, just, you know, be creative, but be sensible.
Okay. There's a question here from Carl Cornish for Steph. Steph, I don't know if you can see this one, but it says, what sawdust did you use? I've been advised not to use oak sawdust chippings because of its high tannin contents as it slows down decay. But you are using oak, aren't you?
I am using oak, yeah. And we have actually found that it seems to have created the right sort of textures. So it has decayed to quite a large extent over the year. But it was dried sawdust because we wanted to make sure that, you know, it didn't have any life in it before. We didn't want to be introducing anything sketchy into these lovely forests. But yeah, I have found that it seems to have worked pretty well, and a large amount of it. I think it goes back to moisture as well. I think if you get the right balance with the moisture, it really helps the decay to move along.
Okay. Matt, another one for you here about if you're looking at a small number of fungi and tree species. What are your thoughts on how this might be applied to other species? Mute. Other way.
Back again?
Yeah.
Yeah, okay. I think it needs to be less about which species you're planning on inoculating, and more about why you're doing it in the first place. What is it you're trying to achieve? Is it are you looking at is it just because you want cavities for bats, or is it because you're interested in a specific beetle if it's veteranizing, or is it a rare fungi that you're interested in? And so the combination of tree and fungi really needs to be guided by that question. So will it work? I mean, it's been trialed, you know, over the last 100 years in 50 odd species, of which there's some evidence for it working. So you totally could use it in any number of trees, but just be guided by why you're doing it in the first place.
Okay. There's a question here from Vicky Wilkins. It's is veteranization becoming embedded in wider forestry management of all woods, forests and parklands, even urban trees, to facilitate natural expansion of the habitat and its associated rare species? So that's to anyone who wants to answer it.
I think I'll jump in here, Ross. Okay. I think it's being adopted, for example, in the Sherwood treescape. I've been working with the Woodland Trust to kind of develop a template by which veteranization could be rolled out over a large landscape area, which would involve different sorts of woodland or tree covered landscapes. So I think there's an appetite for it. Then with regards to urban situations, I think there's always this issue about the balance of safety and amenity and so on. So generally speaking, we've been avoiding doing veteranization where there's high visitor numbers or close to paths and so on, that we're doing that work elsewhere, just because it potentially could lead to kind of conflict of interest there. So, but I do think there's a huge amount of interest for it, and particularly as we're getting more data to kind of to support the efficacy of it more widely than just kind of individual taxa. And it's one of the things that I think is very interesting about veteranization, is it is a kind of cross taxa method, to fungi being the kind of the drivers, absolutely, but also that it's looking like the kind of ecological processes are happening, which is not a surprise because we're matching tree physiology really, and we're understanding tree physiology and using it to our advantage as a tool. So that's not a kind of short answer. It's not a yes or a no, but I think it is on the up.
Okay, thanks all.